Hello again. Today's screencast is this is the third segment for the cell unit, and we're going to be looking at how materials cross the cellular membrane, either leaving the cell or coming into the cell. And you can see here by this microscopic image that we have here, looking at the cell membrane. You can see the cell membrane is a structure that consists of two different layers, and it's referred to as the lipid bilayer, obviously meaning two layers there. And you can see here we have within the cell, you have the inside of the cell, and then you have the outside of the cell here. So we're going to just kind of look at the structures that are involved in this, and also the different types of transportation that occur through this. As you can see here, we're going to look at the cell membrane, and basically what the cell membrane does for the cell, it helps define some of the cellular boundaries, basically what's inside the cell and what's out of the cell. It helps control the interactions with other cells, controls the passage of materials in and out of the cell, and then what is referred to as the fluid mosaic model. And you can see in the image here, as it's kind of shimmying back and forth and moving back and forth, you had this phospholipid bilayer. And a lipid, again, if you remember, a lipid is another term for fats, like oils and grease and that kind of stuff. And your cell membranes consist of two of those, and then inside is the phosphate area. And we have these what we call protein channels that allow for some of the larger substances to travel through. If they cannot pass through the cell membrane itself, it's able to use these protein channels in order to let it travel through. And it's referred to as the fluid mosaic model due to the fact that it's always in motion, and then these proteins give it the mosaic look or structures within the lipid bilayer. And you can see here a little bit more all the different structures that are involved with the cell membrane. We have here, these are what we call our carbohydrates. And these actually act in communication with other cells. The carbohydrates do. They're kind of like an antenna. They do communicate with the other cells. We do have these proteins here, and these proteins are involved in the transportation of some of the large materials in and out of the cell, because most of the cells, most of the materials will be able to travel in and out through the phospholipid bilayer. But in case they can't, they have to use these proteins, and they travel through those depending on what process and what we're moving in and out of the cell. So you can see the fluid, again, the fluid look is because it's kind of moving up and down, and mosaic is because of the proteins and some of these other structures that are found within that. It's going to show us this. So moving on from there, what you can see is the membrane proteins that are involved. And as I mentioned, these proteins are a little bit are going to be used in order, if we have to transport some larger materials. You can see the channel that it's going to travel through here for transportation, some enzyme activity, and also for signal transduction. The main thing that you need to know is what we're going to talk about will be the transportation of these in the of materials in and out of the cell. Again, this is just another point looking at the fluidity of the membrane, how things are always moving back and forth. It allows things to travel through it a little bit easier. In order to do that, you can see how some of the materials here in this particular one, how the cholesterol is able to travel through the cell membrane, not needing a protein channel to do that, and things are able to go back and forth due to the size of their structures. The membrane permeability, and you have seen this in worksheets and your cell quest, a term that is referred to as semi-permeable, or selectively permeable, basically the same thing. So your cell membrane is selectively permeable, or what we refer to as semi-permeable, and what that basically tells us, is it selects for certain materials that can travel in and out of the cell. And what it tells us here is some solutes cross the cell membrane freely, some cross with assistance, and others do not cross at all. So this is what the cell membrane does. It selects for certain materials that can travel through and some that cannot. as you can see in this diagram, okay, so some of these non-charged particles, water molecules, they're able to travel in and out of the cell membrane pretty easily. Some of these macromolecules, or what we refer to as large molecules, cannot. They're too large to slip past the phospholipid bilayer. And then also we have these charged particles. Okay, they are not able to pass through because of their polarity or the charge that they actually have on them. Okay, so your cellular membrane is selectively permeable. Some things can come through, some things cannot. Okay, a little bit more about some of the transportation of the materials through this. Okay, so we have some things that are able to move through. Okay, we have glucose, water, oxygen. These are all molecules that are able to pass in and out of the cell membrane without any assistance. It's able to travel through those phospholipid bilayers. Glucose we know is sugar. We have water, and oxygen is able to do that. Some waste products like carbon dioxide that is also able to do travel through. Okay, the cell membrane. Obviously, the cell, when it goes through respiration, like breathing, it uses the oxygen and gets rid of the waste, which is CO2. Okay, there's two major types of transport processes that occur within the cell. Okay, in the cell membrane, and that you'll have to know. The first one is passive transport. And the second one there you could see is active transport. Okay, we're going to talk about both of them in this screencast that deals with the transportation materials. Passive is just kind of what the term tells you. It's not going to require any energy. And then active transport is going to require some energy in order to do this process. So the first one we're going to take a look at is the passive transport. Okay, as you can see, there's three different types. All right. The key here is there's no energy required. The three different types, we have diffusion, osmosis, and facilitated diffusion. Okay, and osmosis is a type of diffusion, but it deals strictly with water. And facilitated diffusion actually involves a little bit of help, but it doesn't require any energy to do that. And another key here that you should know, that it does always move from a high concentration to an area of low. Okay, so it's going from this high type of concentration that you're going to see. You know, if you want to summarize what this is, high, okay, sorry about the writing here, till low. Okay, and that's the basics of a passive transport. The whole idea is like, for instance, for an example is, you know, if I had cologne or air freshener, okay, and I spray it. When I spray it in the classroom, where it originates from, it's going to be in very high concentration. But what happens is, over time, it's going to diffuse across the room, so everyone's going to eventually smell it. Now, whoever was closer to the source obviously was going to smell it initially. It's at a higher concentration, but it's going to jump from one air molecule to the next in order to get the process of diffusion to occur. Okay, so that's the three different types of passive transport, and we're actually going to go over all of them, and to make sure you have a good understanding of which each one is. Okay, so this is what this diagram here is kind of showing you, is the movement, or what we call diffusion. Okay, so we have here's water on the right hand side, and this kind of dotted line here is what we refer to as concentration gradient. It's just the separation where there's one area has a higher concentration, the other area has a lower concentration, and that concentration gradient is what we refer to. So we have on the left hand side here, as you can see, we have this dye. Okay, it's kind of like if we put food coloring in a cup of water. And so we have a higher amount of water on this side, and what's going to happen is the molecules are going to move this way, or the dye is, and then the water molecules are going to move in this direction, okay, until they reach this point, which we call equilibrium. Okay, so it's going to move from a high concentration to the low. The water is moving in one direction, the dye is moving the other direction, until it reaches this equal point. Okay, now you can also see here, this is where you have two different types of dyes, or solutes, what we refer to. And the orange ones will be moving this way, purple ones will be moving this way, and eventually they will be balanced and they become the equilibrium that we're talking about. And that's what everything's always trying to seek, this balance. Now, once it reaches the balance, doesn't mean it's not going to be moving. Those molecules are always moving back and forth. But once it reaches this balance, the movement's going to equal, and it's always stay or remain the same. Alright, a little bit more diffusion. It's basically, diffusion is the movement okay, of molecules. So we have the movement of molecules from an area of high concentration to an area of low concentration. That's the definition you should know for diffusion. And this is until they're evenly spread out, which we know from the last screen. It is the equilibrium point. And that's the same thing in here. You can see you have this high concentration of dye, or you can even say like Kool-Aid when you dump Kool-Aid in water. And what it does over time, or you mix it up, right, it spreads out until it's evenly spread out. I'm sure before you've had maybe Kool-Aid or something where it's more concentrated in one area and one less in the other, where it tastes like water, when the other one tastes like a lot of sugar. Okay, and that's the idea. It hasn't quite been diffused out yet. You can increase that by stirring it so the rate happens a little bit quicker. Alright, so looking at osmosis, which is basically a type of diffusion, but this one deals strictly with water. Okay, so this is the diffusion of water through a selectively permeable membrane. Remember we talked about that. Your cell membrane is selectively permeable. Some things can go through, some things can't. Water is one of those molecules that can travel through the cell membrane. Osmosis will continue as long as there are more water molecules on the side of the membrane. Okay, and this is what we refer to as osmotic pressure. As long as there's this unequal, there's imbalance, that's unbalanced side, you're going to create this pressure. Now once they're able to balance out, we refer to it as the osmotic balance, is when the inside and the outside are going to be equal. Okay, then we refer to it as osmotic balance. So there's always going to be pressure built up as long as there's an imbalance that's occurring, but once there's a balanced out, there's no longer pressure that's built there. So kind of think of why is it so hard to open a car door if a car ever gets submerged in water? Because of the pressure that builds up. But once you can crack it open and build get the pressure equal, you're able to open the door in the car if it ever got submerged in water. Okay, I actually have a little bit of a slide I wanted to take a look at here with you real quick, okay, that deals with this idea of osmosis. It's pulling up here. Okay, we have that one, and I'm going to also take a look at this one first. Okay, so here we go. Here's water diffuses across the cell membrane by osmosis. You can see what we have going on here. You have a high, or the sorry about that. You have the high amounts of water molecules on one end, okay, and a lower amount on the other. The water molecules are going to move through the cellular membrane, okay, this phospholipid bilayer, through the process of osmosis, going from high concentration to low concentration. Okay, jumping back to our PowerPoint here to keep the notes going along. So you should have all that written down. Now I'm going to go through a few different examples with you. Okay, when you look at this here, we have this, you can see again higher concentration in the dye, the lower amount of dye on this side, but what that tells us is we have more water compared to the water on this side. Okay, so there's more dye, less water. Alright, and what's going to happen then? The water will move out of the cell, okay, or across what we refer to as the concentration gradient. Okay, so this structure here is referred to as our concentration gradient. The water molecules are moving to the left, okay, because what it wants to do is become balanced. It wants to reach this balance point that occurs. Okay, and so it's always seeking to become balanced or the equilibrium. Okay, another example here. You can see, let's say we have our cell. Okay, so here's our cellular structure. Inside the cell is 80% water. Outside the cell it's 100% water. So my question before I put it up to you is, which way will the water be traveling? Will it be going into the circle or the cell, or out of the circle or out of the cell? And remember, we're always traveling from a high concentration to a lower concentration. Alright, well, the water is going to be moving in because inside the cell is a lesser concentration. Okay, and you can kind of see here, just showing you an example here, what would happen to a cell if this occurs and it doesn't become balanced. Eventually that cell can pop or burst. Okay, and then you can kind of think of the plant cell with the central vacuole. As it starts filling up, you water the plant, or the plant, there's water on the leaves. Eventually it can absorb these through the cells and helps fill up that central vacuole. Okay, a second question here is you can look at this, you have 90% water inside, 90% water on the outside. What's going to happen? Alright, so what's going to happen is the water is going to move in and move out, so we're in this balance, and this is what your cells need to have, this osmotic balance that's occurring, so your cells are the perfect size and shape that they want to be. Alright, so you can see that here in this image. And then the third type of cell that could be possibilities here is you can see the cell inside has more water, outside has less water. So which direction should the water be traveling? Of course, it should be traveling on the outside. Okay, and this is what could possibly happen. If this happens too long, the cell will shrink up, kind of like a raisin, okay, and shrivel up. And what you can see here, there's actually a process that occurs in plants. Okay, if this happens, it's called plasmolysis, and this is a term you should know. Okay, this is basically when the water leaves the cell and shrinks in plants. It's called plasmolysis, and you can see here the plant starts shriveling up, everything starts getting smaller, and we'll actually take a look at this in the in class together, and I'll show you an example of this on how it actually works. So by looking at that and going through those couple different ideas that you saw on the three different types of cells, you know, where one burst, one was good, and one actually shrunk up. Okay, these are terms that you should be familiar with: is hypotonic, isotonic, and hypertonic solutions. All right. Now, hypotonic, this is where the cell is going to get lysed, or it's going to blow up to a point where it could burst. Isotonic, meaning it's going to be a normal cell, and you can see here the cell is going to look the way we want it to look. Here's your cell, here's your plant cell. And the third type is hypertonic. Hypertonic means it's going to shrivel up, it's lost its water, all right, and there's the imbalance that's occurring there within your water. Now, this can also happen in your blood cells. And you can see here the different structures. Again, it's kind of opposite of what we looked at in the previous slide. Hypertonic, your cells shriveling up. Isotonic, they're happy. And then hypo, I can always think of when a hypo, meaning it's going to get really big and blow up. So the cell actually will burst like you see here. Okay, and that's not good. When your red blood cells burst, obviously that's what's carrying your oxygen, and it's not going to be able. You're not going to be able to carry enough oxygen to your cells, your other cells, and feed them. Okay, so you need to know the difference between hyper, iso, and hypo, and you should have those in your booklets, the actual definitions for them from previous things that we have talked about. All right, I'm going to skip that, move over to this here, and what you can see here, okay, this particular picture is kind of showing what facilitated diffusion is by having some assistance. This doesn't require any energy, but what you're seeing is this protein channel. Okay, with facilitated diffusion, you might have heard like a facilitator is someone that helps out in like a class or a test or center. Well, in this case, this protein here is assisting. It's still going from, as you can see, from the high concentration to the low concentration, so it doesn't require any energy. It's just that these molecules are a little bit larger. They can't travel through the cell membrane. They have to use these proteins to travel through. Okay, so this is referred to as facilitated diffusion. And I do have a little slide I wanted to show you on that. Yep, there's the bell. Okay, so let's take a look at this real quick. Okay, so here's an image of facilitated diffusion that you can see. A molecule outside the cell binds to a carrier protein. This carrier protein will assist and help it travel inside the cell, okay, and release it eventually when it gets to its point. Okay, and this diagram is in your packet. It's on page 101 in your packet on facilitated diffusion. Okay, back to the PowerPoint. Okay, this you can look at here. This is kind of just going over some of the different types of, this is what we refer to as the sodium-potassium pump, and we don't need to know a whole lot of this. Basically, you just kind of have to know that it exists, the sodium-potassium pump, and it just helps again in the movement of materials in and out of the cell, and requiring a little bit of energy that we're going to take a look at, but the sodium potassium pump, just another idea out there, but you're not going to be required to know it. Okay, so you don't have to worry too much about this one. Alright, looking at this, you can see here we have, I'm sure you guys remember some of the cell parts, and this is just kind of showing you a different type of process that we're going to look at here in a minute on active transport, which requires energy in the bringing materials inside the cell through this particular process that we have here in front of you. So moving on from here is a little bit of summary. All right. Before we do move on to the second type of transportation, which is active transport. Okay. So we've talked about passive. Remember, passive, basically, there's no energy, right? Am I writing on here? Okay. There's no energy required for this. Okay. We're talking about diffusion. Facilitated diffusion, remember, uses the protein channel here. Diffusion, remember, this is the other one that goes along with that, is the osmosis, and that's just the movement of water. We're always going from this area of high to low concentration across the cellular membrane. All right, so we have osmosis, diffusion, facilitated diffusion are your passive transports. Now when we look at active transport, as you notice, it requires ATP, and remember, what makes your ATP is your mitochondria. Okay, so your mitochondria makes that ATP, and it helps us with this process. As you can see, it's traveling through the protein. In this particular case, notice what it's doing. It's going from this area of low or of high concentration, going from a low to this area of high concentration. So I always use the example of, this one here, up here in passive transport, that's like swimming downstream. Okay, we're going downstream. You don't need a whole lot of energy. This is just kind of floating down. But now, when we're talking active transport, if you ever tried to swim upstream or against the current, you're just going to require a lot of energy to do that. Okay, and that's what active transport is. So, active transport requires energy to move substances against the concentration gradient from high concentration to low concentration. So this goes against the gradient from low to high. Okay, you definitely need to know that definition, and you can see that here. All right, so we have our low concentration, looking at the blue balls here, and you have the high concentration here. So let's think, again, think of them as a dye or a Kool-Aid mix or something like that. You drop them into water. Okay, well, these are now going to try to cross the gradient, against the gradient, going from a low to an area of high. So it's like you're trying to shove, you have this container, and you're trying to shove that last substance into that container, even though there's not a whole lot of room to do that. Okay, it's going to you're going to have to use more energy to do it. And again, the red ones are going in this direction. Okay, so it's going from an area of high concentration to an area of low concentration. Okay, there's two special types that are involved in this. Okay, that we refer to as bulk transport. Okay, because what we're doing in bulk transport is we're looking at large amounts of material that are being transported, okay, throughout the cell membrane. And the two different types here are endocytosis and exocytosis. Okay, and you should remember these from our booklets, and when we wrote these words down. Okay, we had endo and exo. Remember, endo means to go in or within. Exo means without, or to go out or outside of the cell. And these are two processes of transporting these large materials in order to do that. So just kind of take a little diagram here and look at these. These are some different types, okay, of transportation of this active transport that you can see. Okay, so we have phagocytosis, we have pinocytosis. And you can see basically what's happening is it's bringing this large substance within the cell. Okay, requires a lot of energy. Same thing here, requires a lot of energy. And the same thing, going in, requiring a lot of energy. And you could see some of the microscopic images here. And in class, I'll actually show you a couple little quick videos that you can see how this actually happens, not live, but how it's been captured underneath a microscope with an amoeba that actually takes in some materials through the process of phagocytosis. So real quick, again, I'm going to show you a diagram that involves this. Okay. Alright, so here's our diagram for this. Okay, so you can see we're talking about endocytosis and exocytosis. The first one here is you can see the materials and the two differences. This first one is endo. So this is when the materials are coming in. You can see the cell membrane slightly opens up here, and eventually it kind of closes off and pinches off, and then the substance is brought within through the vesicles. And then in exocytosis, you have the vesicles surrounding the materials and it's trying to get rid of. Okay, it gets close to the cell membrane, and eventually the cell membrane will open up and let the substance exit outside the cell membrane. Okay, so going back to our PowerPoint and kind of wrapping things up here, that you should basically get out of this. All right, so we've talked about this cell membrane and the transportation materials in and out of the cell. We've looked at the fluid mosaic model. We've talked about active versus passive transport. Okay, active transport meaning it requires energy. It goes from an area of low concentration to an area of high concentration. There's three types. There's your diffusion, osmosis, and facilitated diffusion. Osmosis is strictly with water. Facilitated diffusion uses a protein channel. Okay, and we also mentioned the terms in there: hypertonic, hypotonic, and isotonic. Hypertonic is where the cell is going to shrivel up. Hypo, the cell explodes, and isotonic is the perfect cell that we want. Okay, and then the last thing we talked about was the idea of active transport. And active transport dealt strictly with going against the gradient, so it's going from a low concentration to a high concentration. We talked about two types: endocytosis and exocytosis. Endo, bringing substances within; exo, bringing substances out of the cell. Okay. In class, we're going to run some different demonstrations and discussions to furthermore express the ideas of this and kind of give you a better handle on this stuff. But I hope in this screencast, the third segment of the cell unit dealing with transportation materials across the cell membrane will be helpful and beneficial to you. Make sure you're jotting your notes down, keeping good track of the notes, and there always will be a benefit to you to have the good notes in the 2-column format. All right, have an awesome day.